your first slide, Evan. So Evan's going to talk about automated sampling of patient-derived 3D cell models for metabolite analysis. So Evan, take it away.
Thank you, Terry, and thanks to all the other speakers. Been great sessions and learning a lot today. And my talk will really follow up on a lot of things that Yolanta was talking about. You're using metabolite assays to look at three-dimensional cell models. And sort of as a brief outline, really talk a little bit about the importance of 3D cell models. Speakers before us have talked a lot about this. I won't spend too much time. And then give a little background on our Puma system. It's sort of a unique system in the way we can use microfluidics and flow chips to do sampling and processing of three-dimensional cell models. And then get in some of the data on viability and metabolism in patient-derived tumoroids. And just sort of add a little levity to this. I think the idea that we're now all experiencing assays in 3Ds is pretty exciting. Maybe not as exciting as 1950s movies, but there's a lot of activity going on, and I'm really excited to be part of this. So, and then, from acknowledgements, we've been working closely with Yolanta and Terry and others at Promega in working with their metabolite assays. The patient-derived lines are from our Matt Burrows group at Tulane University. A lot of the imaging work has been done with devices, and then we've done some work with the magnetic nanoparticles, nanoshuttle with Griner. So for the importance of patient-derived 3D cell model, I think Jens showed this same graphic earlier. A lot of precision medicine has been focused on genomic analysis, but now people are starting to realize that you really need to have the complementary information from 3D cell models, and more exactly having 3D cell models that really recapitulate the tumor microenvironment. And that tumor microenvironment is really complex too. And so you have a lot of different cell types, you have the extracellular matrix, you have the different factors in there. And I think the quote from this paper where the figure is taken from is all the cells are contributing to this microenvironment. All the cells are important if you're looking at tumor progression. If you want to solve the cancer issues and get rid of cancer, you really have to address it as a whole system. Now, the group we're working with, Matt Burrows' group at Tulane, they've really developed a number of cell lines from triple-negative breast cancer patients. This is really from the New Orleans area. And usually they work with the patient-derived xenograft, PDX. It was their early work, but now they're moving towards using tumoroids and trying more scalable models. And so a lot of the work we've been doing and the data I'll present is really doing primary cell cultures. So the tumor cells extracted from the patients and then expanded and formed into what we are calling tumoroids. Now, there's a lot of differences in nomenclature, et cetera. And so this is really, but these are primary cells that have been formed into compact spheroids or tumoroids. We're also working with them on doing multicellular formats, multicellular models with extracellular matrix, et cetera. And that's really an expansion of this work. We've been using a multi-cavity device. This is the Agrowell plate from Stem Cell Technologies. Corning has their aplasia plates. There's some other devices that are similar to this. But this device works well for us because we can seed the cells, around 2,000 cells per microcavity. And these microcavities are 400 microns by 400 microns in size. Over a couple days period, then they form into nice tight little tumoroids, and you can collect those and then transfer them into our plates. And so this has really worked well. You can also grow these in single tumoroids and low attachment plates. We've done that a lot. And there's a number of ways of forming these tumoroids. From loading workflow then, once we have formed the tumoroids, we release them. And we can use nanoshuttle. And I think Tim Spicer talked about this, the advantage of having magnetized 3D cell models. In our case, we can use them to transfer and to center them in the bottom. You also use little magnetic pins to pick them up if you want to do single tumoroids, pick in place of those. And so there's a number of different ways you can use the magnetic properties to manipulate and move these around. So it's a very interesting technology. And then at the end of it, this is what you're looking at. So this is the bottom of one of our microcavity plates. You can see the cross section here, 1.2 millimeter diameter across. And with the magnet, it pulls these tumoroids into the center. And so that's really advantageous when you want to do imaging of these devices. You can see the sort of the nice, fairly uniform size of these. In this case, we're getting somewhere 2 to 5 per well. If you want to do single tumoroid, we've also done that. And so you can pick up individual tumoroids and place them into our wells. And so there's a lot of flexibility in our platform. So let me switch now, talk a little more detail about our platform, the Puma system. It's really an automated protocol system that can run in your incubator. So you take preformed organoids 3D cell models. Those are loaded into our flow chips, I'll describe next, along with all your assay reagents. You then place that chip plate into our system, and then there is a pneumatic manifold in there, which is used pneumatic pressure differentials, hence the acronym PUMA, Pneumatic Microfluidic Assay System, to form all your assay protocol steps, all your liquid transfers, et cetera. When that is done, it's now what we call ready for analysis. And so the format is the SLIS ANSI 384 well plate format. So you can go into any imaging device, plate reader, liquid handling system that can work with the standard plates. And then you can do imaging, read luminescence. I mentioned supernatants for ELISA or mass spec analysis. And so it has really a lot of flexibility in how you can then do that analysis. The technology is really based on our flow chip and the microfluidic channel capabilities and layout. So while this looks like a multi-well plate, it's actually a series of chips where you have lanes of test lanes on the chip. And so there'd be 8 test lanes here. The wells in that lane are all connected in the bottom by microfluidic channels. And the central one here, that is a sample well where your organoid or tumoroid would be placed. Unique to our technology is what we call our protected sample chamber. And so the tumoroid would be placed in here, located in that bottom. And then the fluid transfer would come in from the sides. And so you're not getting shear stresses over your tumoroid. You're not displacing, you're not losing it down microfluidic channels. And the volume in here is about a microliter. So you're just displacing 95% of the media. So you can do almost full media exchanges in here without drying out your tumoroid or spheroid or organoid during that process. And this has recently come out in the SLIS technology article. So from that, we can then do what we call multifunctional profiling. So again, the preformed tumoroids are placed in the plate with all your assay reagents. You run your assay in the incubator environment, and then you can do a variety of different tests. You can do viability assessments. You can extract supernatants for metabolite analysis. You can add in antibodies to do staining. And so you can fix the tumoroids right in the chip, add in your primary or secondary antibodies, depending on your staining configuration. And then this all can be done without removing the tumoroid from your flow chip. So once you have it in there, all these steps can be formed and you aren't dislodging, displacing, or damaging your tumoroid. In research areas we've been involved in is a wide variety. Again, because we're an open platform and you can transfer any different type, a 3D cell model in there, you can do things from simple spheroids like HeLa or HepG2 spheroids to complicated microbrains, gastric cancer models. But for this talk, I'll really focus on work we've been doing with breast cancer patient-derived tumoroids and throw in a little bit of melanoma and metabolomics at the end. Okay, so sort of the background of our Puma system, from viability to determine. Again, a little levity here is 3D cell models. Zombies, are they viable? Are these cells live or dead? And if you're doing a 2D cell lawn, it's actually fairly straightforward now with imaging technology to identify each cell, segment it, interrogate, the high content analysis to look for multiple different markers in there. But once you get into 3 dimensions, I think Terry alluded this earlier, it gets much more complicated to do the imaging of those and to be able to collect data and trying to understand where is the cell in that 3 dimensions, or you're looking at 2-dimensional plane. So from this concept of doing multifunctional profiling. Here we're taking tumoroids loaded into our plate, and we can do a variety of measurements before and after the assay. So before, we can do transmitted light inspection, just look for phenotypes of the different tumoroids. And we've also used the real-time glow viability measurements. So I'll show some data from that to look for, ultimately we'd like to do normalization of the number of cells in the well before you begin your assay. During the assay, you can collect your supernatants. You can analyze those from lactate glow, different glow assays or ELISA assays. And at the end, after you've done this, you can then do a viability assessment, live imaging. We also can use cell titer glow, add that reagent directly into the sample well, read luminescence from there as a measure of viability at the endpoint measurements. This is some images of tumoroids that were, again, stained after treatment. And this is using the calcium ethydium homodimer and her staining, added as a cocktail mix into there. These data were acquired with ImageXpress microconfocal systems. So it's a confocal stack of images and then condensed into a maximum projection image. So you're looking at a 2D representation of the different steroids. But from the phenotype, you can see here in the untreated, they remain fairly nice and compact. And then as you treat them, they start to disintegrate, and you see an increase in the number of dead cells as indicated by the ethydium homodimer stain. And so by doing the processing of this and identifying dead cells, you can do a percent viability and get nice drug response curves here, measure EC50 values. This is interesting from the standpoint that the patient was resistant to Taxol therapy, and we can recapitulate that in these tumoroid models. You can see that the paclitaxel EC50 around 10 micromolars, which is over a factor 100 higher than the other two drug compounds. And this is also much higher than you'd see in literature values for other cancer cell lines. And so it is nice to see that these cells can recapitulate, as I said, that Taxol resistance. Okay, so the bulk of the talk then is really focused on these luminescence-based assays. And the two that we'll be talking about next are a real-time glow cell viability assay and the cell titer glow 3D assay. Now, Yolanta spent some time talking about these. So just as a refresher, real-time glow, you have a pro-substrate in your media taken up by the cells. It's, the live cells will churn that out as a substrate, and then you can read that luminescence generated on a plate reader. So we can add this into our well and read the light directly from our plates. You can take it out of our Puma system, read it, put it back in, continue your assay. So it's a very convenient format to use with our plates. Cell titer glow, again, is an endpoint read where you add in the cell lysates and the different reagents and measure the amount of ATP from those. So what we first looked at when real-time glow, and is similar to data that Yolanta showed, was how did the signals vary as you change the size of your tumoroids? And here we're looking at HCT116 cells and just doing different seeding densities in a low attachment plate. And you can see a nice linear relationship between the relative luminescence and the cell seeding density. We also looked at signal versus time. This is looking at MCF7 cells, where we did 2,000 cells per well, again, in these low round bottom plates. And then looking at the response of the real-time glow over time. Now, it takes about 2 hours to establish or equilibrium signal from this assay. But after 2 hours, you can see that it's very nice stable. So our bottom line is we're seeing a nice linear response with size and a stable response with time. And so very encouraged about using this as a normalization tool for 3D cell models. Moving now to tumoroids. Again, looking at tumoroids, different number of tumoroids, because we're using the same cell number to generate these, we increase the cell number by increasing the number of tumoroids. And this is actually more realistic from our device, where you might have multiple tumoroids at the bottom. So, can we distinguish between 1 and 5 tumoroids? And you do get a nice, fairly linear response here with the number of tumoroids. This is a 384 well plate where we can control better the number there. And then we also looked at in flow chips And this is where we would place a single tumoroid in a flow chip, and then looking at the signal to background we're getting here. And so we get, a nice signal background 44. And if you look at the standard deviation of the background, the signal noise around 690. So very nice measurement capability for doing single tumoroids and looking at their viability in our flow chips. And then last is endpoint viability measurements. Again, here we're using cell titer glow, introducing these cell titer glow reagents into the sample well, churn up the cell, look at the luminescence, and you can see a nice concentration dependent decrease in viability. In parallel, we can do the viability using staining, and you can see the increase in the red stain, the completed homodimer, and sort of nice correlation actually to the concentration response of the other traits. So again, just showing the different type of measurements possible in our flow chip, doing luminescence-based cell titer glow, doing confocal imaging. You just mentioned real quickly, we can do really high resolution imaging because we have a very thin bottom on our plate. So it's a thin COC bottom compatible with really high resolution imaging. Okay, so the third part is really cell model metabolism. And we'll pop by here showing how you can generate energy through different compounds, in this case, spinach. And the importance of metabolism in cancer is talked about, by some of the previous speakers. But I think the Warburg effect is one of the more common things. This is from the 1956 paper by Otto Warburg, where he is really showing that normal cells go anaerobic glycolysis, but when the cancer cells, they start undergoing aerobic glycolysis. And you see an increase in lactate production there. But really, the importance from oncology, I think, is looking at metabolism as a target. Can you alter metabolic pathways as a therapeutic strategy in attacking cancer? And if you really want to understand how that works, I think it's important to do dynamics, that you can't just look at the endpoint of it, because the cells are going to be dead and they're not going to be metabolizing. But if you can look at that intermediate steps, and we'll show some data, you can really look at disease modeling. How do cells respond to cancer treatments? How do metabolism responds? I think it's a very exciting field. So we use the lactate measurement here. This lactate assay, again, shown by Yolanta, allows you to look at single spheres. In this case, looking at tumoroids and look at number of spheres per well, the type of signals we're getting. So very sensitive. It actually works really well with our system because of the small volume that we have in our wells and the small volume required for this assay. I want to talk a little bit then about how we're doing the sampling. So the standard way you would see for people doing these type of metabolite and looking secretions, you put a larger number of spheres into a well, and then you take out a small volume from that. So for example, removing 10 microliters from the 300 microliter volume. And you can do that multiple sampling without decreasing volume too much. That gives you what we call integrated metabolite response, where if you load this up with glucose at the beginning, then over a certain time period, glucose will decrease, your lactate will increase, and you can measure that, typically measure as an endpoint, but you can also do multiple, measurements in between. For our system, because our fluid exchanges a complete volume, so we're doing a one-for-one exchange of conditioned media with fresh media, you get what we call discrete metabolite response. And so if you're looking at, for example, glucose, you're taking a smaller amount of glucose out, and so you see a smaller decrease. But if you're looking for a secreted factor such as lactate, now you're actually doing what we call a flop-in experiment, is you're looking for an increase above zero background or very minimal background. And so then if you model this as signal to background, again, for the glucose, which is being depleted in this model, in the integrated discrete, you can measure that, and integrated is going to give you a better measurement of the depletion, but you can still do that with discrete measurements. But if you look at the secreted factors, in this case lactate, which is being produced by the glycolysis, you get a much higher signal of background when you're looking at discrete sampling for secreted factors. And so in that case, I really think our device, which allows you to do that discrete sampling in an automated fashion over multiple time points, really affords a very nice tool for studying metabolism dynamics. So the workflow that we developed then, this is again working with Promega and their glow assays, is you would load your tumoroid and media components into the Puma system. At preset times, you can then do a one-for-one exchange of fresh media or fresh media compounds with your sample well. And at the end of it, you generate, in this case, 5 different time points. We have our baseline measurement, which is media only. And then we have 4 different time point measurements over a 12-hour, actually 15-hour period here. So each sample is around 20 microliters. With the dilution factor, you have plenty to run multiple different analytes and do different repeats in 384-well plates. And so it's a very nice correlation. So from one flow chip, which has 32 samples, you can generate up to 6 or 7 time points per sample, and giving you over 200 different data points for an assay, and really allow you to really map out the metabolism dynamics. So here's some data. Again, these are from tumoroids, and sort of going through the plots here. We're looking at response of individual tumoroids over different time points to different concentrations of these compounds. And the pictures at the bottom show you the endpoint viability staining of those representative tumoroids. As you can see at the end, we still have live tumoroids. This is a 12-hour treatment, and so the concentrations weren't high enough to kill the spheroids, but you can definitely see effects on the metabolism. For example, paclitaxel at the 9 and 12-hour periods, you see a definite increase in lactate production, potentially as it's trying to respond to the treatment and respond to the drug compound. And trametinib, maybe less response, but at 12 hours, you see an increase again of that. So this is something that Matt's group is looking at much more closely. Very intriguing data, really being able to look again the metabolism dynamics, see how does the cells respond to treatment, and how does metabolism respond to treatment, and can that be used as a target for therapeutic treatment. So the last set is a little shift. It's still metabolism related. This is metabolomics. And because we were doing this fluid exchanges and everything in the incubator, you can now do what we call in situ sampling, in situ lysing of samples. And so from a metabolism standpoint, if you want to see what the metabolism profile of a tumoroid is, the current way you would do it is you would take it out of your incubator environment, you'd put it on ice to try to freeze metabolism, and then wash it and process it, lyse it, and then run that on your mass spec. In our system, you can do that without removing the tumoroid from the sample, so without perturbing its environment. And so I think that's going to be critical for these type of work. First, we want to see what sort of sensitivity could we get from single tumoroids, in this case, single spheroids, HT116 spheroids with about 2,000 cells per well. Because we're lysing in the small volume, get the natural concentration increase. And here we're able to see around 250 different metabolites from these cells. And if we did a little experiment here where we treated them with different compounds, staurosporin and C59, which is the Wnt inhibitor, you see nice grouping of the different spheroids, from the control, which actually came out tighter than we expected, and the staurosporin and the C59 treatments here. So it's a really nice proof of principle that you could really use our system to lyse single tumoroids and look for metabolomic profiles and changes in the profiles as response to treatment. And then a group at UT Southwestern, Sean Morrison's group, they take this and they're looking at now melanoma tumoroids, again, using different type of treatments and seeing good grouping of the treated versus control groups these weren't single tumoroids, there are multiple tumoroids in there, but we think we can push this to single tumoroid measurements for them too. Okay. So just in summary, I think we're trying to emphasize the importance of patient-derived tumor models. Again, is echoing some of the previous talks, how important 3D cell models are for not only oncology research, but all sorts of other type of biological research and development of therapeutics. The incorporation of those with the excellular metrics and recapitulating the tumor microenvironment is important. The work we're doing right now is a more simplified, but we're extending that to extracellular matrix. So we can do this with hydrogels, with matrigel. We can do it with multicellular components in there. And then I think ultimately combining this with genomic profiling for the precision medicine. From our system, I think where we really offer advantages is as an open system, allowing you to do these fluid transfers and supernatant sampling in situ, doing automated and time course sampling, and really perform different type of assay workflows. Showed a little bit of immunofluorescence staining, but it's another area that we find a lot of use for our system. And finally, just doing this multifunctional profiling, so being able to look at different aspects of viability, metabolites, functional assays. I think the luminescence-based assays that Yolanta was talking about and that we've been using from Promega are really nice, elegant solutions and provide that single tumoroid sensitivity and allow you to do that in dynamic process. I didn't emphasize it, but by dynamics, we are collecting the data in 2 to 3 hour time slices. Again, if you let the tumoroids secrete for overnight, you're going to get a lot more material. But with the sensitivity of these luminescence assays, you can really think about doing 30-minute, 60-minute time points, and you collect enough secretion from those in discrete sampling that you can really map out the dynamics of metabolism and response of cancer models to treatment. So with that, I'd like to say thank you. And if there are any questions, I'll be happy to answer them. My contact information if you want any more information on our system.
Great. Thanks, Evan. Thanks for that, I should say, illuminating presentation. I don't see any questions yet in the Q&A box, but I do have a couple myself.
Okay.
And it has to do with the heterogeneity among spheroids. This is just a general question. It could probably be handled also in the panel discussion. And that is it better to use multiple spheroids per well? Or is it better to use multiple wells, each with an individual spheroid?
I think it depends on the question you're asking. If you want to look for heterogeneity, if you're doing imaging, you can, in some sense, separate the tumoroids in the well, and that allows you to look at that heterogeneity. And we've done some of that, especially with these 4IC cell lines. You see a lot of different response of tumoroids coming from the same cell population to treatments, and so there's definitely a lot of heterogeneity. I didn't talk about because we really haven't quantified it too well, but there's an indication that we're seeing more variation in the real-time glow and these luminescence assays from the 4IC cells than you see from immortalized cell lines, which would say that just the heterogeneity of these and the responses is important. But from a metabolite, I think you really want to do single tumoroid and look at secretion from a single tumoroid if you want to look at the heterogeneity. So.
Yeah, and I think there's the opportunity to normalize to certain other parameters as well. I think that will help deal with the variability. Another thing, just a comment that I saw, you showed some graphs that indicated linear relationship with the number of cells seeded and RLU values, but they didn't. I mean, there wasn't a direct relationship, and I think a lot of people see that and have a question, but if they think about it, the physiology of cells change when they're in a more crowded environment in the spheroid. So that, I think, shouldn't be too much of a concern for people. It shouldn't be, I guess a way that they misinterpret the data.
Yeah, not only the physiology of the cells, but you have the penetration of the reagents. If you're looking at secretion, how are those coming out? And so there's Yeah, there's a lot of different factors to take into
Yes.
account as you're optimizing for 3D models versus nicely, controlled flat 2D cells on plastic.
And again, I'm not an expert in imaging, but I'm still always challenged when I see the reconstructed models. And when you see an image of a spheroid and they're looking at something, oftentimes there's a periphery that's stained, some color. And my question usually is, why isn't that in the middle? I mean, if you're seeing a full projection image and you're looking at both the top and the bottom of the spheroid, which should be the same as the edge, why isn't it brighter in the middle instead of absent in the middle?
That's one of the dirty secrets of what you call a maximum projection. So for each plane, you'll take the pixels that are in best focus, and you'll eliminate the pixels that are out of focus in those planes. And so in some sense, it tends to give you just the outer surface, but you can get some of the inside. But yeah, the maximum projection is more about producing the coolest image you can get, as opposed to all the data you've collected from that three-dimensional model.
Yeah, okay. Do we have other questions from the audience? The participants?
Ladies and gentlemen, if you'd like to ask questions for Dr. Cromwell, please kindly raise your hand electronically. We will unmute you, or you could type your questions in the Q&A box or in the chat. And that's also a way for you to engage with Dr. Cromwell.
So one thing, Terry, just along that line is, there's the image you present, which shows you sort of a representative image of that 3D cell model. From the analysis standpoint, we typically will go, image by image to do the analysis. And so you're doing your live dead analysis off each image plane and identifying cells in each image plane to calculate live versus dead.
Yeah. Whereas
from the presentation, then you just show the maximum projection image, which is sort of a collapsed version of that whole image stack. So.
Okay. Okay, I don't see any others coming in. I think what we'll do is we'll move to the panel discussion and open it up for general questions to begin with.